Okay, so today we're going to try something new. Instead of doing regular notes where I'm in front of the class, you're just going to watch this video recording of the PowerPoint. This allows you to pause at any time. You can rewind. You can take as much time as you need. So that way we can also pause it to take you out and have discussions about your science fair project and review some of Unit 2. Preferably use headphones, but if you do not have any, keep the volume down so that this isn't disturbing anyone else. What you're going to need, just like any other set of notes, you're going to need a pencil or pen, your catalyst sheet, you're going to need Friday's exit ticket that you picked up from your folder, you're going to need notes number 17 on mitosis, you'll be working on the homework, which will be the next 2 pages in the packet, and the mitosis web quest. So just like you would normally see, you have the date, you have your I can, you have your agenda, your catalyst questions, and the homework that you need. So feel free, you can pause right now, you can copy these things down, answer your questions, feel free to use your notes from Friday. But yeah, pause, and then whenever you're ready, you can play again. So going over the exit ticket from Friday, number 1, why is it important for DNA to be duplicated exactly before a cell divides? Hopefully at this time you have your exit ticket out. If you think you have the correct answer, if you had D, that was it. Basically, we need to make sure that the DNA, the genetic information, is exactly perfect so that each new cell gets the right amount. Number 2, why is it important that the 2 sides of a DNA molecule are held together by weak bonds? Going over your answer, the correct answer is again D, because those DNA strands are opening and closing and opening and closing. We need to make sure that those are weak bonds so that they can be broken easily. We're going to use each one as a new template. Number 3, what is the complementary DNA strand to the following? If you had this DNA half of a strand, it would match up with these complementary bases. And just so you remember that, apples in the tree, cars in the garage. A goes with T, C goes with G. So what we're going to be talking about today is mitosis, which is talking about how certain organisms can regenerate missing limbs. So for example, the starfish here, it lost a leg and it grew it back. Same with the gecko. If it loses a tail, it can regrow it. However, we cannot do that. If you lose your hand, you cannot grow it back. However, what we can do, we can heal up cuts. If you've ever cut yourself or scraped your skin, you notice it's not a scab forever. It grows back, makes new skin cells. And again, this is how plants grow. This is how you grow. This is how things go from a teeny tiny organism to the thing that it is today. So you started out as a teeny tiny baby. Now you are a growing adult. And this is all because of the cell cycle. Usually we just shorten it as mitosis, but the whole thing is called the cell cycle. And there's 4 different parts that the cell will have to go through when it grows and divides. So we start with the G1, right there. Move on to the S phase, the G2 phase, and then the M phase, M for mitosis. And we're going to explain what each of these 3 things mean, 4 things. So step 1, we have the G1 phase. The G stands for gap, and what happens here is the cell is going to get a little bit bigger. It's going to start making new proteins, making new organelles. It knows that it's about to split, so it needs to make sure that it has enough things to pass on to the new cells, the 2 new cells. After we have our G1 phase, that's where the S phase comes into play. S stands for synthesis, and this is where DNA replication happens. This is what we talked about on Friday, where you go from 1 strand to 2 strands. You are replicating that DNA, that complementary DNA. We have the G2 phase, and again, that G stands for gap, and this is the last chance. It says, are we ready for mitosis? Did we have everything ready? Did we make enough proteins? Did we make enough organelles? Did our DNA replicate properly? I like to think about this as like you're about to go on a road trip. This is like, did I pack my toothbrush? Did I pack my toothpaste? Did I go to the bathroom one last time? This is the one last step before we do mitosis. Okay. And again, our last phase is the M phase, which is broken down into two different parts. And those two parts are mitosis, when the nucleus is actually dividing, and then it's followed by cytokinesis, which is the cytoplasm and the cell membrane is actually splitting. We're going to go into each of these parts way more in depth. But the way I remember cytokinesis, it's all the C's: cytokinesis, cytoplasm, cell membrane. And again, looking here, we have G1, S, and G2. All of these things together we just call interphase. Most of the time in the cell cycle, the cell will be in interphase. It's getting ready. So it's growing, it's making sure it has enough proteins, enough organelles, didn't replicate its DNA properly, and it's making sure it's ready to go. Because if not, this is how you can get diseases such as cancer or other cell mutations. Alrighty, but thinking about this, why does a cell have to go through interphase before it divides? And again, interphase is these three parts right here. So why do we do that? You're thinking, and it's because we have to make sure that there's enough cytoplasm, enough proteins, enough organelles, and a full set of identical DNA before splitting into two identical cells. We also have to make sure that it's actually ready to divide. Did we replicate this properly? Do we have enough organelles for the new cells? So this is why we do G1, S, and G2. And when we have our DNA, it's not always going to be the same depending on which stage it is in. So in interphase, you can see that, oops, you can't see my mouse. During interphase, over on the left-hand side, the DNA is in a loose form called chromatin. During the S phase, DNA is replicated. So now you see it goes from one strand to two strands. Each of those strands is called a sister chromatid. And then once we actually get into mitosis over on the right-hand side, that is going to be your chromosome. And what happens here, it's going to be nice and tightly coiled. It's nice and neat and organized. So this would be like, if you talk about chromatin, that's your headphones, you just put them in your pocket. It's going to kind of be a mess. However, in a chromosome, that's if you actually take it and wind it up and tie the ends together so it's nice and pretty and nice and neat and organized. I know that we're not doing this together, but this will help you practice. So get out a board, grab a marker. After each of the questions, just pause it, draw what you think the answer is, play it, and that way you can see if you're right or not. So if you were looking at DNA in interphase, pause. All right, in interphase, the DNA would look kind of like a mess like that. It's in that chromatin form. However, what would it look like during mitosis? This is when it's in its chromosome form. It's coiled up, it's nice, it's neat, it's organized. During which stage of the cell cycle does DNA replication occur? Again, pause while you write this down. So DNA replication occurs in the S phase, and S stands for synthesis, when it's synthesizing that DNA. What is interphase? Pause. Interphase is G1, S, and G2. So it's all of those things together. It's getting ready to divide the cell. What stage does the cell spend most of its time in? Pause. Again, just like we talked about, it would be interphase, spending most of its time there, getting ready to go. What two steps occur during the M phase? Again, pause. If you said mitosis and cytokinesis, you are correct. Mitosis is the division of the nucleus. Cytokinesis is cell membrane and cytoplasm. Now, if we break mitosis down even further, this is when we go from one cell to two identical cells. And there's 4 different phases that we're going to talk about. And feel free to pause to copy all this down, but I'm going to go through each of the phases. Interphase, this is before mitosis, but just keep in mind it's G1 followed by S followed by G2. Once we actually get into mitosis, we start with prophase. Pro means before. This is when our DNA condenses into chromosomes from chromatin. And also the nuclear envelope is starting to break down. This is the one that everybody always forgets about, but the nucleus starts to break down as well, so those chromosomes can move. After prophase, we have metaphase. Think M for middle, M for metaphase. They're going to line up in the middle of the cell, the center of the cell. They might also say the equator of the cell, but that's just the middle. And also we're going to have these spindle fibers attached to the chromosomes. I like to think of those as little lassos, so it's going to actually latch on to the chromosome, because we're going to need that for anaphase. And in anaphase, think A for away or apart. What's going to happen is those chromatids, those 2 little arms, they're going to split right down the middle, and they're going to go to opposite ends of the cell. So anaphase, chromatids are moving away. And then finally, we have telophase. This is when the chromosomes are going back to chromatin, and the nucleus is going to start to reform. And the easy way to remember this is PMAT. Prophase, metaphase, anaphase, telophase, PMAT. And then don't forget, after we do mitosis, after we have those 2 separate nuclei that you can see right there, we have to do cytokinesis so that we actually split the cytoplasm in the cell membrane. Again, if this is kind of complicated, feel free to rewatch what you've just seen so you can review this again. Now, why in the world do we actually do mitosis? This is a form of asexual reproduction, so you only need one parent to make identical cells. It makes 2 daughter cells each round, so you start with 1 cell, you end up with 2, and they will be identical. And this is used for growth. This is also used for repair. This is how we make identical cells. So again, if you cut your skin, it will grow back. It will make skin cells, not stomach cells. They're identical to the parent. And again, I'm going to trust that you are writing on your boards. So once I read the question, pause it, write down your answer, and then play it for the correct answer. So if we are doing this, how many cells does mitosis make? How do the daughter cells compare? You can draw a picture to represent this process. So hopefully you wrote down, we're making 2 identical cells to the parent. You can draw that if you want, but it goes from 1 cell to 2. During which stage of mitosis does the nuclear membrane reform and the cell prepares for division? So pause. This is telophase. Remember, the cell is dividing in cytokinesis. And this is when the nucleus is reforming around those chromatids. During which stage of the cell cycle do the cytoplasm and cell membrane split? Pause. Remember, all of these start with C, so it must be cytokinesis. And then what are the 4 stages of mitosis in order? Pause. Hopefully you wrote down PMAT and then added in the rest. So PMAT for prophase, metaphase, anaphase, and telophase. And then what happens before mitosis? Pause. If you said interphase, you were correct. We have to make sure that we're ready to go. We have to replicate our DNA. We have to make sure our proteins and organelles and extra cytoplasm have been made. Which stage of the cell cycle is depicted? If it depicts mitosis, number the stage 1 to 4 according to when it occurs. So looking at this, pause, this is interphase because it's that loopy mess of chromatin. This one right here is showing what? Write it down, pause. This would be anaphase, number 3 of PMAT, because the chromatids are being pulled apart. What is happening here? This would be step 2, metaphase in the middle. I forgot about prophase. Yeah, metaphase in the middle. This right here is showing what stage? Pause. This is telophase. We have 2 nuclei in this case, so this is the 4th part, the T of PMAT. What stage do you see right here? Pause, write it down. If you said prophase, that is correct. So you see it went from chromatin to chromosomes, but there's no rhyme or reason to where they are. They're just chromosomes. They haven't moved to the middle yet. This right here is what? Pause. If you said cytokinesis, that is correct. You see we have 2 separate cell membranes that have been formed. All right, so hopefully that made sense. Hopefully you practiced. If not, re-watch this video. But what you are doing right now is while we are still working with you, you're going to be working on the Mitosis web quest. That's the individual paper you picked up. You'll go to the websites at the top of each section and look for the answers. You may work with one partner once you are both completed with the notes video. If somebody is still working, get started on your own. They can join you when you're done. The next 2 pages of your homework, which I will be checking tomorrow, is the classwork Mitosis. Look at the title if you're confused which 2 pages they are. Any missing work you might have, check that progress report, see if you have any zeros, any missing assignments, and then your exit ticket as well. If I have not talked to you yet, I will be talking to you about your science fair project, so be prepared for that. If you need to work on any parts of that, feel free to do that as well. But other than that, get to work.